considering in general terms the way that information is decoded from genes to produce proteins. And as we know, genes are segments of a piece of DNA, which is part of a chromosome, and there are different portions of the long molecule of a chromosome are devoted to particular genes. And each gene encodes a particular protein that consists of amino acids that fold into a particular shape, which dictates a protein's function. So in more specific terms, we know that DNA is transcribed into RNA. I'm only showing one strand of DNA here, the DNA that is the same as the RNA, the coding strand of DNA. And the triplet code that is present in DNA and RNA is translated into individual amino acids, with each triplet codon specifying a particular amino acid in a protein chain. And then it is the type of amino acids, their order and their number, that dictates how a protein will fold and what the protein's function will be. So now it's time to look at transcription and translation in some detail. And we're going to start out with prokaryotic transcription and point out some major features of prokaryotic transcription. So we're looking at transcription in bacteria primarily. There is a single RNA polymerase enzyme responsible for transcription of bacterial genes, unlike eukaryotes where there are 3 different RNA polymerases. And in prokaryotes, there's the initiation of mRNA synthesis does not require a primer, unlike DNA synthesis, which, as you remember, does require a primed template. RNA polymerase does not require a primed template. It simply requires the appropriate an appropriate stretch of DNA, which can then be transcribed directly into a single strand of RNA. And the RNA polymerase requires a promoter sequence, a start site of transcription, and a termination site of transcription. And together, these elements that are found in the DNA compose a transcription unit, a unit which will be transcribed into a messenger RNA. So we're going to look at these in sequence later. Let's talk about the promoter first. The promoter does not encode amino acids in a protein. Rather, it forms a recognition and binding site for RNA polymerase, for the enzyme to bind. And it is found upstream of the start site of transcription. It is not transcribed itself. It simply binds RNA polymerase and orients that polymerase so that it will transcribe a gene, an operon in bacteria, a sequence of genes. And the promoter is asymmetrical in that it has a polarity to it that indicates the site of initiation downstream from the direction of transcription. And I'd like to pause here to point out, when we talk about transcription, we need to get our directionality straight. So if we have a stretch of DNA here, and we will say this is the start of transcription here. This is going to produce a messenger RNA. This is the start site of transcription, and as we know, only one of the 2 strands of DNA will be transcribed into a complementary mRNA. And we refer to that site as the +1 site that represents the start of transcription. So +1 will mark the site at which transcription occurs, and as we will see later, a translation start will occur somewhat downstream of the transcript initiation site. And so this region here is a untranslated region. It will not be translated. So there's a region of the mRNA here between the start site of transcription and the start site of translation, which is untranslated, an untranslated region. And when we speak of genes in both prokaryotes and eukaryotes, we can speak about the polarity of the gene. And since RNA, like DNA, is transcribed in the 5' to 3' direction off a DNA template, which runs in the 3' to 5' direction, this strand of DNA would be anti-parallel 5' to 3'. Since the direction of transcription of mRNA is 5' to 3', we can refer to this region of untranslated region as the 5' UTR, the 5' untranslated region. And we speak of sequences upstream of the transcription unit that would be in the 5' direction from the start of transcription. So this would be upstream. And the promoter in bacterial systems, the promoter is found upstream of the transcription start site. And there are two regions, as we will see, that are very important for the promoter. There's one region of DNA sequence at minus 10 nucleotides upstream from the +1 start of transcription. And there's another region of the promoter in the DNA that is found at minus 35 nucleotides upstream from the +1 start site. And both of these regions are contacted by RNA polymerase protein prior to the initiation of transcription. And the promoter, the point that we're making here is that the promoter is asymmetrical in that it dictates that transcription will start downstream of the promoter, not in this direction, not upstream. So it will direct transcription to start at a transcription start site about 10 nucleotides downstream from the minus 10 promoter site. And that is where transcription will be initiated. So in terms of the polarity of the gene, we could refer to this as the 5' end of the gene or operon in bacteria, and this as the 3' end. There's a polarity, and that polarity is given by the direction of transcription of the messenger RNA, 5' to 3'. So that's very important to consider when we refer to different DNA sequences that are in different positions relative to the transcription start site at +1. Now the polymerase itself in bacteria is, we refer to the entire polymerase as a holoenzyme, meaning the entire enzyme. And it is composed of multiple subunits. The core enzyme has two alpha subunits, a beta subunit, and a beta prime subunit, whereas there is an additional subunit, the sigma subunit, shown in purple here, that it dissociates from the core enzyme as after initiation of transcription. And it is the sigma subunit which gives specificity to the RNA polymerase. And there can be several different sigma subunits in bacteria. And when a particular sigma subunit is turned on or expressed, it then associates with the core enzyme and dictates which genes, which operons will be transcribed by the RNA polymerase. So if we look at the binding of RNA polymerase, we see that it contacts the promoter region at minus 10 nucleotides upstream from what will be the transcription start site here. And it also contacts the DNA of the promoter at minus 35 nucleotides upstream. And you can actually map these binding sites on the DNA and see that the protein actually makes contact there. We won't go into those experiments. If anyone wants to talk to me outside of class, I'd be happy to talk to you about those footprinting experiments. So one of these strands is we refer to as the template strand. That is the strand that will be transcribed. And the other strand of DNA we refer to as the coding strand because it has a sequence of nucleotides that is just like the messenger RNA that will be produced, because the messenger RNA is complementary to the template strand, and the coding strand of DNA is complementary to the template strand. So the coding strand of DNA looks like the messenger RNA, except that it has nucleotides that contain thymine instead of nucleotides that contain uracil, as in the RNA. So the sigma subunit binds to the DNA, and the helix, the DNA double helix, opens up at the minus 10 sequence. And what happens there is that a transcription bubble is formed, and with ATP, with the hydrolysis of ATP, the sigma subunit dissociates from the core enzyme, and the enzyme then begins transcription at the +1 start site of transcription, 10 nucleotides down from the minus 10 sequence of the promoter. And Then elongation ensues. The core enzyme minus the sigma subunit, which is dissociated from the core enzyme, that enzyme moves along the template strand, synthesizing RNA in a 5' to 3' direction exclusively. Just like DNA polymerases, RNA polymerases only synthesize polynucleotides in the 5' to 3' direction. Now, the transcription bubble at initiation and during elongation contains the RNA polymerase, the DNA template, and the growing RNA transcript that is being made by the addition of ribonucleotides to the growing RNA chain by the RNA polymerase. And after the transcription bubble passes a stretch of DNA, the transcribed DNA is rewound. So it looks something like this, schematically speaking. We have a transcription bubble here. This is the coding strand in that it will look like its nucleotides will be the same as the messenger RNA, because the messenger RNA that's going to be synthesized in the 5' to 3' direction as the RNA polymerase moves in this direction. We are synthesizing 5' to 3', adding new nucleotides to the growing 3' end of the messenger RNA. The messenger RNA has nucleotides that are complementary to the template strand, and the coding strand has nucleotides that are complementary to the template strand. And we can say that we can refer to the region 5' in the 5' direction from the direction of transcription as upstream sequences, and sequences downstream from the initiation of transcription, and using the polarity of transcription 5' to 3', we can refer to sequences that lie 3' to a growing transcript as downstream from the growing transcript. So again, these terms upstream and downstream, you should understand, refer to the directionality dictated by the 5' to 3' transcription of the DNA and the production of mRNA in the 5' to 3' direction. And so the RNA polymerase moves downstream as it transcribes the template strand of DNA into messenger RNA. And as a transcription bubble is formed, and as the polymerase moves down the transcribed DNA, the transcription bubble closes behind this complex, and the double helix is rewound, it reforms. So the transcription bubble is only a small region of single-stranded DNA that allows the polymerase to transcribe the template strand. So we have unwinding of the double helix ahead of the addition of nucleotides to the growing RNA strand. And then as the transcription bubble passes a region of DNA, we have rewinding of the double helix. Now, in prokaryotes and bacteria, there is a termination sequence that's part of the transcription unit, and that signals the RNA polymerase to stop transcription. And it causes the formation of phosphodiester bonds, that is by the mechanism of nucleotide addition, which is very similar to the mechanism of nucleotide addition in DNA synthesis, and you can review that if you'd like. We are adding nucleotides to the 3' end by having a nucleophilic attack of the 3' oxygen of a ribosugar of the last added nucleotide onto the alpha phosphate of an incoming nucleotide. And as the termination site is reached and we cease formation of those phosphodiester bonds, that is, we stop polymerization, the RNA-DNA hybrid within the transcription bubble dissociates so that the DNA is released and the RNA transcript is released then, and the DNA rewinds. Now, essential to the action of the termination site in the DNA is the formation of a hairpin in the RNA as follows. So if this is a termination site where we have 4 or more U-ribonucleotides and a particular sequence that will form Watson-Crick hydrogen bonding, this stretch of RNA made as the polymerase transcribes the template DNA to the termination site, this stretch of RNA will form what we call a stem and a loop. We call this a hairpin. And that hairpin causes the RNA polymerase to pause and dissociate from the DNA releasing the mRNA as a messenger RNA that will then be can be translated into protein. Now, in prokaryotes, because there is no nucleus, we can have transcription of a sequence of DNA into messenger RNA, and immediately, even prior to the cessation of transcription of a stretch of DNA, translation can begin. So the messenger RNA begins to be translated before transcription is finished, in other words. And in bacteria, the transcriptional region that we often refer to is an operon. So as opposed to the transcription of single genes that we will see in eukaryotes, there's transcription of multiple genes in one messenger RNA. Those multiple genes are clustered in an operon. So we have a transcription of an mRNA that actually contains the information that will be translated into multiple proteins. That is, we have what would be called in bacteria, we have a polycistronic mRNA in prokaryotes. This is in prokaryotes. And all polycistronic means is that it's polygenic. A cistron is another word for a gene that, for historical reasons, we call it cistron. So the messenger RNA that emanates from transcription of a bacterial operon has a 5' untranslated region, 5' UTR. It has a start site of transcription, and then there will be a start site, this is start of transcription, and then there will be a start site of translation. And then there will be a stop site of translation. And then depending on how many genes are in the operon, we will have another start site of translation. And then there will be another stop site of translation. And there could be another start and another stop of translation, such that the start and stop regions of translation, let's mark them off here. There's one start and stop of translation, another start and stop of translation, and here I've drawn a 3-gene operon in bacteria, third start and stop site of translation. Each of these will be translated into a different protein. Protein 1, say, protein 2, say, or protein 3, say. So this is the nature of the polycistronic mRNAs that are caused by the transcription of operons. Together, this entire unit would be referred to, along with its promoter, as an operon. And so operons contain multiple genes, and they often encode multiple enzymes for a pathway, for a biochemical pathway. That is, you find clustered together in a single operon, which is transcribed into a single messenger RNA, the genes for proteins that are functionally related in that they have a common purpose in the cell. They are often found as enzymes in the same biochemical pathway. And later on we'll consider operons that encode enzymes for catabolic pathways and operons that encode enzymes for anabolic pathways, anabolic and catabolic pathways. We'll talk about both of those when we talk about gene regulation. So the advantage of clustering genes together in a single operon, which is transcribed as one unit and then translated into individual proteins, the advantage is that these protein levels can be regulated together. And when we get to the regulation of gene expression, we will see the elegant mechanisms that come into play to regulate whether or not an operon is transcribed or not. That is, whether the enzymes encoded by an operon are made or not. And we will consider that a little later on. So the important point here, one of the important points here is that prokaryotic transcription is coupled to translation. And we can actually see that in an electron micrograph. And this is the schematic interpretation of this electron micrograph. What we have here is an operon in bacteria that is being transcribed by RNA polymerase molecules. So the red arrows mark RNA polymerase molecules here. And as the RNA polymerase moves along 5' to 3' synthesizing mRNA in the 5' to 3' direction, it is elongating and messenger RNA, which grows longer and longer the further along the operon as transcription proceeds. But simultaneously with the transcription of the messenger RNA, we have ribosomes that associate with the messenger RNA and ribosomes that are elongating proteins, that are manufacturing proteins, that is creating polypeptide bonds linking amino acids together into polypeptides that are directed by the nucleotide sequence of the messenger RNA. So we have coupled transcription and translation in prokaryotes. There is no nucleus that separates the transcription of genes in the nucleus, as in eukaryotes, from the translation of messenger RNA in eukaryotes by ribosomes in the cytoplasm. Instead, in prokaryotes, we have coupled transcription translation because there's no nucleus separating the ribosomes from the DNA which is being transcribed. So we see that in prokaryotic transcription, we have a promoter, which RNA polymerase binds to. We have a transcription start site, about 10 nucleotides down from the initial binding site of the polymerase to the promoter. We have multiple genes that are transcribed in a single mRNA, a polycistronic messenger RNA, in other words. And we have a site of termination that is dictated by a particular DNA sequence that signals the RNA polymerase to stop translation. And we have coupled transcription and translation. Those are the major features of prokaryotic transcription. We'll talk a little bit more about operons later. Now it's time to move on to eukaryotic transcription. And unlike prokaryotic transcription, there are 3 different RNA polymerase enzymes in eukaryotes responsible for transcribing different types of genes in the eukaryotic genome. So we have RNA polymerase 1 enzyme, which transcribes the ribosomal RNA genes into ribosomal RNA. And as the name implies, these RNAs are components of the ribosome, as we will see. The ribosomal RNA is a very important component of the ribosome. The ribosome consists of both ribosomal RNA and proteins. So together, RNA, ribosomal RNA plus multiple proteins make up the ribosome. And the ribosome is the protein manufacturing machinery of both prokaryotes and eukaryotes. And ribosomes are what we refer to as ribonucleoproteins because they contain both RNA and proteins. So RNA polymerase 1 transcribes ribosomal RNA genes in eukaryotes. RNA polymerase 2 transcribes messenger RNA and some small nuclear RNAs. And for most of the protein coding genes in the eukaryotic genome, those are what produce mRNA because it is messenger RNA that is read into protein on ribosomes. But there are some small nuclear RNAs that are involved in RNA splicing that are transcribed by RNA polymerase 2 as well. That is, there are some genes that encode snRNAs that we'll talk about when we get to splicing that are transcribed by RNA Pol2. So we refer to these as Pol1, Pol2, and finally Pol3, polymerase 3, transcribes transfer RNA genes into transfer RNA, and we're going to study transfer RNA in great detail, as well as some other small RNAs. So whereas in prokaryotes, the ribosomal RNAs, the messenger RNAs, and the transfer RNAs are all transcribed by the same RNA polymerase, these three classes of genes are transcribed by different RNA polymerases in eukaryotes. And each of these different RNA polymerases has its own type of promoter sequence that it recognizes and binds to. So for example, the promoter for a typical protein coding gene would have specificity for RNA polymerase II binding, whereas transfer RNA genes would have a promoter that is specifically bound by RNA polymerase III. So each polymerase recognizes its own type of promoter and therefore initiates transcription of its own type of gene. Now, in eukaryotes, the initiation of transcription requires a series of what we call transcription factors that are needed for a eukaryotic gene to be transcribed. So the situation is much more complex than in prokaryotes, where we're looking at essentially a holoenzyme consisting of a sigma subunit plus a core enzyme. Here we require multiple transcription factors to load RNA polymerase at the promoter necessary to transcribe a gene. And we're going to focus on Pol2 genes and the RNA polymerase II transcription of genes. And Pol2 requires a series of transcription factors that we'll refer to as basal transcription factors, and then we'll also consider auxiliary transcription factors as well. Now, these basal transcription factors interact with the RNA polymerase to form an initiation complex at the promoter, and they are required. These transcription factors, these basal transcription factors, are required to load the RNA polymerase effectively at the promoter. Now, unlike prokaryotic transcription, the termination of transcription in eukaryotic genes is not as clearly defined by DNA sequence. Basically, there's no set termination point. The polymerase will eventually fall off the DNA and release a messenger RNA for transcription. And unlike prokaryotes as well, we have monocistronic mRNAs in eukaryotes. That is, in eukaryotes we have simplified, we'll have say a double-stranded DNA here, and this will be the start of transcription for a particular RNA. This will be the start of transcription. And like prokaryotes, there will be a 5' untranslated region downstream from the start of transcription, and then we will find a start site for translation. And eventually there will be stop site of translation that we will consider later, because in eukaryotes, this mRNA, we would call this a pre-mRNA, and this will be processed. So we'll cover that later. That's another difference between eukaryotes and prokaryotes. So far we have a promoter like that, similar to prokaryotic transcription. We have a promoter to which RNA, in this case, we'll consider RNA polymerase II binds, and it will initiate transcription downstream from the promoter. But we will have a single messenger RNA that will produce a single protein eventually. After processing and translation, we will find one protein. So we don't have multiple genes in one transcription. We have one mRNA for one protein. And we'll talk about this RNA processing and how we get to one protein a little later. So termination is not well defined in eukaryotes as opposed to prokaryotes. And so schematically, in the schematic drawing, what we have here is a transcription factor that binds to a region of the promoter, the eukaryotic promoter, which we call the core promoter region, which contains what we call a TATA box. And the TATA box is a DNA sequence to which the transcription factor binds. This is called TF2D, transcription factor 2D. And there are other transcription factors that associate with TF2D. And when this complex forms, RNA polymerase can be loaded at the core promoter, forming an initiation complex. And then RNA polymerase, after initiation of transcription begins, it will move down the template synthesizing a messenger RNA. So the loading of RNA polymerase II in eukaryotes requires a series of basal transcription factors to be assembled at the core promoter region. And the first contact occurs at the TATA box with a transcription factor called TF2D. And then these other basal transcription factors associate with that and load the RNA polymerase and get it ready for transcription. So it's a little more complex than that, and I'm going to show you this schematic and go over the mechanism by which RNA polymerase is activated to transcribe a eukaryotic gene. So down here we will have the start of transcription. You can see that RNA polymerase is loaded onto the core promoter when we have a series of basal transcription factors that assemble in response to the initial binding of a TATA binding protein called TF2D to the TATA box region of the core promoter. This is called a TATA box because it has a TA repeat in it in the DNA sequence. So it's very important that you understand that when we refer to a promoter, we're referring to a DNA sequence to which, in this case, in eukaryotes, basal transcription factors are assembled and which recruit RNA polymerase to that core promoter. So the promoter is a DNA sequence, not RNA nor protein. And the RNA will be initiated, the transcription initiation site will be downstream of the core promoter. Now, what determines, or what helps determine whether or not these basal transcription factors are assembled at the TATA box has to do whether activator transcription factors that we can call auxiliary transcription factors, positive auxiliary transcription factors, bind to sequences of DNA which can be very far upstream and sometimes even downstream of a gene. And these activator transcription factors bind to regions of DNA that we call enhancers. And when they bind to enhancers, they can assemble co-activator proteins. There are a lot of co-activator proteins that help recruit the TATA binding protein, or TF2D, to the TATA box and help recruit these other basal transcription factors to the core promoter, which then loads RNA polymerase, which then can begin transcription. So whether or not RNA polymerase begins transcription of a gene in a eukaryotic system depends upon the binding of activator positive transcription factors that we call, well, I'll refer to as positive auxiliary transcription factors, to particular DNA sequences that we call enhancers. So we have promoter sequences, which are DNA sequences, enhancers, which are DNA sequences to which positive transcription factors bind and enable the assembly of the complex, the initiation complex at the core promoter, which allows RNA polymerase to begin transcription. And there are also silencer regions of DNA to which negative transcription factors bind. And often these negative transcription factors occlude or block the binding of positive transcription factors to enhancers. So we have silencer DNA, we have enhancer DNA, we have promoter DNA. And it's important that you study these and become familiar with these terms, because if we are to understand eukaryotic transcription in detail, we'll need to refer to this again when we get to the regulation of gene expression in eukaryotes. So now we're ready to tackle messenger RNA modifications that occur in eukaryotes. And before a messenger RNA is produced that can be translated in the cytoplasm, we've got to process, in other words, the primary transcript that comes from a eukaryotic gene. And that's where we'll pick up with in the next lecture.